And then I think there was someone yesterday who actually described all the ANI and all the nuances of the new names, HIV-affected brain disorders. There's a decrease in viral load to undetectable, and it totally makes a difference. And by the way, I'm not going to go into it, but I think it makes a difference, not because the virus is neurotropic, but because there's less activation in the periphery. There's a decrease in this neurotoxicity, because, again, decrease in virus. Encephalitis, though, Eliezer Masley at UC San Diego, he still detected about 25% in HIV encephalitis at autopsy, which was really surprising. That was about 10 years ago. And the other thing is the complications of aging impact cognitive impairments, so that including cardiovascular disease and renal disease. So these complications are tough. And in my group, we study mainly older HIV-infected people, so these complications do come into play. So the evolution now, we've got to a chronic infection. So people are treated. You have a decrease in the CD69 on the monocytes, a decrease in CD16, a decrease in TNF. And then you have this mild impairment, which makes it a little harder to study because we know what caused that flaming encephalitis. Now what causes this very subtle change? And so it makes it a little harder to study. So how does a monocyte macrophage look after all these years and on ART? In my lab for the last, I don't know, probably 15 years, we've been doing mRNA arrays. And so we do this on monocytes from patients with HIV because we want to see how they respond. What's going on in the periphery, the monocytes responding to. So we just take whole blood, and everybody's probably done these things before. We magnetic beads, we purify CD14 monocytes, and then we do mRNA arrays. And I published several of these over the years to see how they've changed. And we were funded a few years ago to look at HIV-HCV co-infection, because it turns out that a lot of the papers that were coming out, and Dana Kabuza actually published one showing that there was a lot of dementia, even now, and it just so happened that they didn't mention it, but a lot of them had HCV. They were co-infected, which is why I asked the question yesterday if someone had a lot of HCV co-infection. So we were funded to look at HIV-HCV co-infection and their activation, and before and after therapy now, because now people are getting treated. So the co-infected people we studied have undetectable HIV because they're on ART, and they have high loads of HCV because they haven't been treated yet, but they will be. And so what you can see here is we looked at HIV undetectable, HIV viral load, again, this is all message, these are patients, co-infected, HCV alone, and then controls. The co-infected, remember, is HIV undetectable viral load, and they really should look more like this, but they don't. They have a very high, what I look here is interferon-induced genes. People with high viral load and HIV have this pattern that we've published, and they have mainly the highest expressing genes. All of these are interferon-induced. And then if you look over here at the HCV, these genes right here are all HCV associated. But if you look here, these are different than these. Turns out these are LPS-induced genes that we don't see that often here, except we start to see them with the high viral load, but not so much with the low viral load. But the co-infected people are looking very much like HCV and a combination of HIV with high viral load. So what we did is we looked for one of the interferon genes that we could measure in the plasma, and one of them was IP10. And we wanted to know if the gene expression correlated with the plasma, because we were looking for biomarkers, maybe for cognitive impairment. And it turns out that there's a really good correlation with IP10 concentration in the plasma and the gene expression. So we did find this to be a nice correlation. This is HIV, by the way, not co-infected at this time. But it didn't correlate with cognitive impairment. So we wanted to know what other markers. So we did a large study, and we included MRS. So we did MRS neuroimaging and found that the IP10 in the plasma correlated with a decrease in N-acetylspartate, which is considered to be a marker for neuron damage. And you see a very nice correlation here. So we did show that the interferon at least seemed to correlate with the damage in the brain. We also looked at the brain sections of people who died of HIV and found that the SN, which is CD169, it's also called sialoadhesion, was greatly increased marker on monocyte macrophages. And here you see perivascular macrophages, CD68, and a subset are staining with SN. So we knew that there were interferon-expressing monocytes getting into the brain. And now I'm going back to the co-infected. And again, we compared the co-infected with the HIV. Here's co-infected, HCV. We did 7 neuropsych domain testing and came up with a global deficit score. And a global deficit score of greater than 0.5 is considered to be impaired. And what you can see here is that the co-infected, we had about 60% of them that were cognitively impaired, which was surprising to us. The HIV is obviously still significant compared to the co, and then a co, of course, is compared to the control. And then what about the LPS? We noticed that the LPS levels were elevated, and they were certainly elevated with the HIV undetectable, even though we didn't see much on the monocyte. So we have a monocyte in the HIV people, at least, that have high interferon, but we have circulating LPS, so we can't really ignore the LPS that we see. We're not sure that the monocyte's responding to it anymore, but at least in primary alone HIV infection, we know HIV has LPS levels. But the LPS levels, it's surprising to us, they don't correlate at all with the global deficit score. So it's not the LPS we think that's doing anything, but we started to look at LPS because we think it contributes to this interferon and that the combination is synergistically bad. So this is co-infected. We took the top genes. There were 6 top genes overexpressed. 5 of them were interferon genes. 1 of them was an HCV-associated gene. And what you see here are the dots are co-infected, and the triangles are undetectable HIV. And you can see that as the activation goes up, we came up with an activation score of these genes. As activation goes up, cognitive impairment goes up. And then as you can see here, the activation is not that high anymore with these treated undetectable HIV, but they're still impaired. Okay, so now I want to go into something new. We wanted to know, is there another mechanism for this impairment? So 2013, the Nobel Prize was for these 3 gentlemen who identified that there was vesicle trafficking coming off of all cells, and that it was a major transport system and a cell-to-cell communications. These were called microvesicles. So, exosomes. Exosomes are one of the microvesicles. There's usually 2 types, and it's become way more complicated, so there's actually an article out this month even talking about more differentiation between these vesicles. They're usually based on size and where they bud from, either the plasma membrane or endosomal budding. The endosomal budding is considered to be an exosome if it is coming from the endosome and also 70 to 100 nanometers in size. So we were going to, and all of the hoopla was mainly exosomes at the time. Exosomes can be identified by these tetraspanins that they have on the cell surface. There's CD81, 63. They also on the cell surface, they have sphingolipids, lipid rafts here. Inside, they're really abundant with miRNAs. They have mRNA. We're not sure it's functional. We're still trying to decide what's actually inside these things. But most of the research in this, and I went to a meeting 3 years ago, the first international extracellular vesicle meeting, and 98% of the people were from Europe, and most of the work was on tumor cells, because it was fascinating to think that the tumor cells could bud these exosomes and then spread the exosomes around the system and cause metastasis. It was considered to be the latest thing in metastasis, for certain, especially glioblastomas, if any of you have been reading the literature. It was glioblastoma exosomes and the spread. How do you isolate exosomes? You take whole blood. In our case, I wanted to do monocyte exosomes. You take whole blood, you culture it overnight in exosome-depleted media. That's really important because there are exosomes everywhere. So fetal calf serum has exosomes in it. So you use exosome-depleted media. Then you add this, in the past, they did ultracentrifugation. It takes a long time to do this. It has evolved where companies now have rapid ways to isolate exosomes. We use ExoQuick TC, which is tissue culture. To clarify the supernatant, we incubate overnight, and then you get exosomes in a centrifuge pellet. Now you can freeze these exosomes, and you can analyze them later. We isolated RNA from the exosomes by Kaisal, and then we looked at, in particular, miRNA. So this is a characterization of the exosomes. This is how people have to show this to show that they are exosomes. You look by EM to say that they're uniform. You do a nanoparticle tracking analysis to show that they're between 70 and 100. And then you do at least one, if not 2 or 3 tetraspanons to show that you have exosomes. Here we have monocytes alone. Here is this exosome tetraspanon, CD63. Oops. And so they're on our exosomes. They are not on our monocytes. So we have pure exosomes. And then we decided what we would do is we're going to look at the exosomes and we're going to activate the monocytes. So we're going to activate the monocytes, isolate the exosomes, and see what they look like. So what we do when we need lots of blood is we use leukocyte reduction chambers. We get them from the blood bank. And then we enrich for monocytes and we put them in low attachment plates because we want them as monocytes. And then we treat them with interferon, LPS, or a combination. I call it IL. We do interferon first and then LPS. And then you incubate them, you culture them exosome-free media, and you get the exosomes. So we've now treated, whenever I say this, I'm talking about exosomes from monocytes that have been activated. Now, exosomes are very high in miRNA. So we did miRNA arrays on normal monocyte exosomes. We did it from HIV, HCV co-infected people. So we did 10 co-infected people. And then also, we didn't do macrophages because we didn't have time, but we did use what was in the literature, miRNA on macrophages from this publication by Ismail in Blood. So the most abundant miRNA in these microvesicles or exosomes is 223, MIR223. So the gray bars are monocytes. The dots, the blue dots are macrophages, and the red stars are from the patients. So I want you to focus first on the monocyte and the macrophage. The monocyte and the macrophage had the same miRNAs. It's just their abundance is slightly different, but not much. If you look at this, 90% of the MIR is 223. That's huge. So just look at the line here. Even though there are differences, even there are differences here in some of these, you're talking about 3 and 2 and 1%. So monocyte and macrophage, very similar. Macrophage, for example, MIR goes down on some of these MIRs, up on some of them. So it is a little different, but not much. And then the patients. So this is where we're going to focus our work. The patients themselves have an increase in certain MIRs over a normal monocyte. So we might be looking at some of these MIRs that are in the stars. And then it's really important to know, does the monocyte have the same MIRs as the exosomes? So we were interested in these activated. These are the ones that are the most abundant. 223, 146, A and B. And we added 155 because it's been shown to be part of activation, and it's associated with activation, even though it's not that abundant in normal monocyte macrophages. We treated the monocytes, we activated them with the normal activators, interferon, LPS, and combos. And then you can see here, this is the fold change after activation. And most of the fold change is with the LPS and the interferon LPS combo. But the take-home lesson I want to show you is that the exosomes are pretty much showing you the same thing that's in the monocyte. So that's pretty cool, because that means that you can have these exosomes and they can be put into anything else, and they're almost exactly like a monocyte. So, can exosomes enter other cells and functionally deliver cellular cargo? So first we decided that for the monocytes to get into the brain, you need to go through the endothelial cells. And so we wanted to look at endothelial cells. So unfortunately, these are HUVECs, and I need to do the brain endothelial cells, although I do expect them to be similar. So we labeled the exosomes with this exo green, and then we added them to HUVEC cells. And what you see here is the HUVEC cells, and then these are just gobs of exosomes entering the HUVEC cells. And then if they enter the HUVEC cells, do they do anything? And so what we wanted to do is to look at the ones that are activated. Again, these are activated. And look for P65. Look for nuclear translocation of NF-kappa B, just to see if anything's happening. The interesting thing is if you look at the non-treated, so these are non-treated exosomes. So they're like just monocyte exosomes that normally come out. And then you've got down here no exosomes. So these are just, HUVECs with no exosomes. They don't do anything. This is normal communication. Then when you do the interferon-activated monocyte exosomes, you get this real increase in the cytoplasmic P65. And if you look at it, it's not really nuclear. This is cytoplasmic, and I'm assuming this is probably some kind of protective mechanism. LPS, surprisingly, you don't see the P65 translocation. And we've done this like 4 times. We've submitted this for publication. You don't see P65 translocation. But the combination, you start to see that these are all nuclear translocation. So we knew the combination was doing something, and it was probably synergistic, and we wanted to know what. So the most important thing I would think of for endothelial cells would be maybe activation of a chemotactic factor. And indeed, that's what it is. These are Western blots. We did CCL2, and we've also done ICAM, which I wasn't going to show you because I didn't want to run out of time. But they have very similar reaction. So when you add these exosomes, you get a 67% increase in CCL2 just with the exosome from the activated monocyte. And this is just the Western blot and the quantification. And then what about secretion? We looked at secretion of IL-6, TNF, and CCL2. So these are just ELISAs. Again, these are activated. Here are the activations here. And then here they are. TNF doesn't do anything. It seems to be an IL-6 reaction, and the CCL2 is secreted. And we see the same thing with ICAM. So just for those of you who may not be familiar, there's an awful lot of information coming out in the last year on exosomes and neurotoxicity and all of these conditions, especially because they're showing exosomes can carry the A-beta protein. They can transport alpha-synuclein, PRP proteins associated with neuronal exosomes. MS is a little tougher. It says it's protective and destructive function. So there's papers on both sides. Okay, so now what about neural cells? And so this is what we're working on now. We wanted to take the activated monocyte exosomes and put them in neural cells. These were normal ones. We wanted to see if they even get in. So normal monocyte exosomes, not treated, not activated. Primary astrocytes, these are all human. Primary astrocytes, primary neurons, here they are, and then here are the exosomes. What we see is they probably don't get into neurons quite as well as astrocytes. We've repeated this several times. They seem to be like sucked up by the astrocytes, not so much with the neurons, but it does get in. And then finally, again, we wanted to see is there damage, with these treatments, the treated monocyte exosomes. Again, so we did P65. And in this case, we added LPS as a control, positive control, and we do see translocation, not too much. Again, these are non-treated or no exosomes. They don't do anything. So normal monocyte exosomes don't hurt astrocytes. LPS treat, this is LPS directly. We just threw LPS on astrocytes. And so you do get something. Again, the interferon seems to have this cytoplasmic P65. The LPS, we do see more, but then we start to see, again, a synergistically really bad effect on astrocytes with nuclear translocation, and also the morphology seems to be distorted. So those are studies that we're doing right now. The HUVEC studies are the ones that have been submitted. Oops. Okay. And so I'm going to finish with this cartoon. And I think maybe to give you all some ideas, I think you can take the monocyte macrophage, again, monocyte periphery, and you can activate it with microbes or autoimmunity or anything in the periphery. And then what we're showing is you can certainly activate endothelial cells, and then you can have the exosomes cross the blood-brain barrier by themselves, let alone have the monocyte cross over in the macrophage. And then we know that these exosomes can get into neurons, oligos, microglia, and astrocytes. And then each one of those cells, so it's like a cascade. The monocyte can go into an astrocyte, let's say. The astrocyte can produce distorted exosomes, and that can go into a neuron. I mean, it can get pretty complicated, so we're trying to do it a little step by step. But they all will shed, and there's literature now to show that they all shed exosomes in the central nervous system. And then the question is, do those exosomes come out of the central nervous system? So I'd just like to acknowledge the people in my lab and Dr. Monto, who's the clinician who helps recruit patients, and also my funding from NIMH. Thank you.